Okay, so this is lesson 1.2, part 6. So let's get started first by going through the practice questions on incomplete dominance from the last lesson. Okay, so we are almost finished section 1.2. So these are from page 41 and 42. So remember that in some of the older documents, it may show just 2 capital letters. My expectation is that you use the capital letters for the gene and the superscript for the allele. So that's kind of why I've crossed out and written on top of those in the question. And this applies to our assessment, so you should be using the notation that you were taught. Okay, so we are crossing a red CR carnation with a white CW carnation. So we can see here that this CR parent is donating one allele to the offspring and the CW is donating the other allele. So the result here is all offspring are the genotype CRCW, meaning they are 100% pink. Example 2, a red CRCR is crossed with a pink CRCW carnation. So again, this first allele is donated from the parent on top. This one is donated from the parent on the side, and so on throughout. So we can see that 2 of the offspring are CRCR and 2 are CRCW. So 50% are red, which is the CRCR, and 50% are pink, CRCW. So again, it may be helpful to start with the genotype before the phenotype. On the test. This is how the questions will be set up. Your Punnett square may also be different in terms of ordering, and that is okay. So just be aware and make sure your genotypic and phenotypic ratios match what I have there. So example 3, a pink CRCW carnation is crossed with a white CW carnation. So we can see, looking at our first Punnett square, that we have 50% are pink, and that's these ones right here, and the other 50% are white. So 50% are CRCW, making the pink, and 50% are CW, which is white. Number 4, a pink CRCW carnation is crossed with a pink CRCW carnation. So in this case, we get 25% CRCR, which will give us our red phenotype. 50%, which again are these 2 right here, give us our CRCW phenotype, which is pink. The last genotype is CW, which gives us our white phenotype. So that's 25% or 1 in 4. Next questions. Is it possible for 2 red carnations to produce anything other than red carnations? No. As you can see from the cross, there are no CW alleles present. There are only CRs, so they will all be red. Number 2, is it possible for 2 white carnations to produce anything other than white carnations? Why or why not? Similar to number 1, but this time they're all CW, so they can only produce white offspring. Number 3, is it possible for 2 pink carnations to produce anything other than pink carnations? Why or why not? Yes, pink carnations can produce red, white, and pink offspring. So this is example 4 that we just looked at. Number 4, what phenotype does each parent need to be to produce only pink offspring carnations? Why? So 1 red and 1 white would need to be present. So all offspring will get 1 red allele and 1 white allele from each parent. Why do we refer to this as incomplete dominance? Because red does not completely dominate over white. It mixes together to produce a pink. So this is incomplete dominance. This is incomplete because in complete dominance, that's a little bit confusing wording wise, the red would be completely dominant if both alleles were present. So we would never get pink. If it was codominance, we would see spots, i.e. both colors would be visible. Again, no pink. So in order for it to mix to produce that pink, it needs to be incomplete dominance. Okay, so this link for the Amoeba sisters on incomplete and codominance, you can stop at 4 minutes in because after that it goes into some other stuff, is linked here as well as on Teams. So just watch the first 4 minutes, which covers incomplete and codominance, if you're still a bit confused and want more information. After 4 minutes, yes, it moves on to something else, so you don't have to worry about that unless you're interested. Okay, so now we are going to be going into sex-linked traits, which is page 36. So in humans, we have 22 pairs of homologous chromosomes and one mismatched pair, which are the sex chromosomes. So at least they're mismatched in the male. So we're going to look at some traits that are specifically tied to the sex chromosomes, so the 23rd set. So genes for these traits are located only on the X chromosome, not on the Y chromosome. So for an example, let's say we have a gene located here on the first X chromosome. First off, before we get into that, it's good to note that the Y in this example is not actually that big. As you've seen in karyotypes, the Y chromosome is quite a bit smaller than the X, and it's also less of a Y shape than it is shown here. It looks more like a mini or funny shaped X in reality. So if you have a gene on this arm of the X chromosome, which I have circled here for you, and it's for a recessive trait. So think normal dominance, recessive, not codominance or incomplete dominance here. So if you have a dominant gene in the same corresponding spot on the second chromosome here, so the second X chromosome, I should say, it will be hidden. So the trait will be hidden and we won't see the gene being expressed because it's being masked or hidden by the dominant allele on the other chromosome. If this recessive gene is in the same spot on the male X chromosome, so right here, instead of being paired with another X, it's paired with a Y. And there is no corresponding gene because if you look here, the section is missing. So there's no section of DNA there. There's no gene there that could block the recessive gene or mask it or hide it. So the recessive gene then gets displayed or expressed. So you do not need a second copy of the recessive because there is no one dominant at all present to hide it. There's only one chromosome that it can be shown on. So X-linked alleles always show up in males, whether dominant or recessive, because males only have one X chromosome. So if recessive is in the female, you would actually need 2 copies of the recessive trait for the gene to be expressed the same as in males. But in the male, the sex-linked or X-linked allele, which is the recessive allele in this case on the X, will show up no matter what because they don't have another X to dominate or hide the trait. So females are therefore much less likely to be affected by recessive X-linked traits because it's very unlikely that they would have 2 of the recessive alleles, but they can be carriers for that recessive allele. Because females have 2 X's, one of them could be recessive, meaning that they would carry the recessive trait without actually expressing it. Okay, so some recessive sex-linked disorders. So examples of recessive sex-linked disorders, again, are more likely to show up in males than females because males only have one X chromosome. There's no way to carry the recessive trait on their one X chromosome. It would have to actually express it. So color blindness. There are different kinds of color blindness. Just so that you are aware of that, it's not just one thing. But color blindness is the inability to distinguish the differences between certain colors. Most common type is red-green color blindness, where red and green are seen as the same color. The allele, for example, N for normal and little n for color blindness, is attached to the capital X, representing the X chromosome. A capital Y is used to represent the Y chromosome. So we don't show an allele on the Y because there is no corresponding allele on that Y to block the recessive trait. So notice that there is no allele on the Y. Okay, so this top image is an example of a color blindness test that you might take at like an optometrist's office, for example. So if you have red and green color blindness and you were to look at this image, you would not see the 29 present within the circle. So this QR code here, I would like you to take a moment and try because it's pretty cool. Even if you know you're not colorblind, it's still worth it to try. But this runs you through a color blindness test that tests for all the different types of color blindness. So pause here, give this a try, and then you can come back to your notes. The QR code is also available in your notes. So in that QR code test, you'll get examples similar to these types of images. Some of them in the test are fake to ensure you're doing the test properly. Some of them are to distinguish between different types of color blindness. If you are colorblind, you might see the wrong number or no number present at all. Okay, so let's try an example here for color blindness. So a female that has normal vision but is a carrier for color blindness. So she has a normal gene as well as carries the color blindness allele, marries a male with normal vision. So again, no colorblind allele present. Give the expected phenotypes of their children. And then we've just identified that capital N is normal vision, little n is colorblind. So if you're a female carrier, you obviously have two X's. One will have normal vision allele, one will have recessive colorblind allele. So therefore you are a carrier. So this woman does not have color blindness, but she does have the allele present. The male has normal vision, no allele present there for that one. So we're going to use the Punnett square shown here. So we've got our female up top. Okay, so we have the mom and the dad, or the male and the female. So looking here, we can see that phenotypically, so if a capital N is present anywhere, that phenotype will be normal vision. So we have 75% normal vision, meaning that the remaining 25%, or the 1 out of 4, is colorblind. So all females in this example, you can see the two X's have normal vision, and 50% of the males, so out of the two that are there, 50% of them were colorblind. Okay, another example is hemophilia. So another sex-linked disorder. So again, this is more common in males than females. Hemophilia is when the blood won't clot properly and a small wound will bleed incessantly. So what that means is, for example, if you were to get a paper cut, it would bleed more than the average person without the condition. But a more serious cut, because obviously a paper cut will eventually stop, but a more serious cut would keep bleeding and cause major problems. Again, with hemophilia, females are more likely to be carriers than they are to actually have the disease. So if we did a cross, this would look really similar to color blindness. So we can see that first off, from our sex inheritance, there is a 50% chance of a child being male or female. And then from that 50%, if we had 4 kids, we can see that we would have a son with a normal X from the mom, meaning that they don't have hemophilia, a daughter who would be a carrier as she inherited the allele for hemophilia from her mom, but she would be normal, she wouldn't have the disease. Then there would be potential for a son who has hemophilia, so he got the affected X from his mom and the Y from dad. And then there's a daughter who got a normal X from dad, a normal X from mom, so it does not carry the trait. So it is also good to know the image in the corner here, the lower corner, is that hemophilia is actually a very famous example of a family tree for Queen Victoria's bloodline. So in the past, royals would marry other distant relatives to keep the bloodline royal. So through this inbreeding, it resulted in more women being carriers for the recessive trait, resulting in more offspring, including women who then had hemophilia, because the dad would have hemophilia and the mom would be a carrier, meaning that the daughter would then have the condition as well. So what does this look like in a Punnett square then? So a female that has a normal blood type is not a carrier for hemophilia, so she's X capital N, marries a male with hemophilia, X little n y. Give the expected phenotypes of their children. So to do this, we are again using a Punnett square. So the female is here, who is not a carrier, and the male is affected by the condition. So looking at the table, we can see that for the genotype, 50% are X capital N, X little n, meaning they're a carrier for hemophilia, and 50% are X capital N, Y, because the mom is normal and she is who is donating the X chromosome to the sons, so there is no chance that the sons could have the affected X chromosome. So the phenotype then is 50% female carriers, meaning they have the allele, but they're not expressing the hemophilia disease, and 50% normal males, which means that 100% without hemophilia. So none of the offspring will have hemophilia because they all have at least one normal allele for this condition. Okay, so a non-human example is calico cats for sex-linked traits. So only female cats can have 3 fur colors: black, red, and white, aka known as a calico cat. And this is because the black and orange color is a trait that is sex-linked, where white is controlled by another gene. So black could be a male or a female cat, and this is the same with the red or orange cat. But the calico must be female, as it has to have 1 black X chromosome and 1 orange X chromosome. So the male can only have 1 chromosome, 1 X chromosome. So they can only be either black or orange, not both. White is independent from that. Black and orange are co-dominant, so that female can show the 3 colors. So that is our last example for this lesson. So I would like you to work through page 44 and 45, which has questions on sex determination and sex-linked genes, and then work through these before moving on to the next video.